Tēnā koutou katoa. Ko kurahopo te waka. Ko tapuai o unuku te maunga. Ko wairo te awa. Ko rangatane te iwi. No te whanganui a tāra aho. Ke karori tōku kāinga. He kai pūtaiao tōku tungamahi. Ko rawiri ekuru tōku ingoa. Nō reira, tēnā koutou, tēnā koutou, tēnā tātou katoa. Kia ora. My name is David Eccles, and I am a theoretical geneticist working here at the Milligan Institute of Medical Research. I want to talk to you today about a side project that I've been working on over the past few years, relating to the visualization of DNA of arbitrary lengths. I've talked about some of this before, but that's very much in line with the subject that I'm talking about, which is repetitive DNA. Now, the image on my title slide has been used frequently by the Milligan Institute for publicity purposes. It's a panorama photo, and it's actually a crop from a full 360-degree panorama photo that you can see here. Made from 16 source images, which I stitched together using a free software program called Huggin. For those who aren't familiar with photo stitching, in order to assemble the panorama, you first identify control points for matching bits of different images. The ideal points are things that have high contrast and are easy to identify in the other images that you're using. There's a bit of an art to a good panorama. You need a good eye for the repetitive structures that can cause problems, including the things that look locally unique, but are duplicated in completely different regions of the big picture. If you're lucky and have control of the camera, then you can create source images that make sure that no pictures sit entirely within repetitive regions. So there's always a unique bit in each image that can be matched to neighboring images. But I'm not here to talk about panoramas. I'm here to talk about genomes and DNA. Another one of the metaphors that I quite like about DNA is a book, or more correctly, a library of books. A typical book will have on the order of 100 to 1,000 pages, each filled up with letters in a particular sequence. That makes up effectively a linear sequence of a few 100,000 to a few million letters. The very start and ends of each book look quite similar, and if you think of them in terms of their appearance, then you could say that the front of a book is a little bit like the reflection of the back of the book. Some books are grouped together based on some loose connections that are not always obvious to casual browsers of the library, and they can be held together in sturdy bookcases that are simple and sometimes relatively uniformly painted and stained. Now, when DNA is represented at an atomic level, we see that it is composed of 4 different nitrogenous bases supported by a sugar phosphate backbone. We call these bases adenine, guanine, cytosine, and thymine. A fundamentally important chemical property of these bases is that they have a unique natural partner that is one of the other bases. Each DNA base is able to pair up with its natural complementary partner via hydrogen bonding, forming a strong but reversible bond that allows 2 chains of bases to combine into a very stable structure. Due to the physical constraints of this bond, the backbones are oriented in opposite directions, creating 2 related sequences that we call a forward sequence and a reverse complement sequence. Okay. So short double-stranded chains of DNA sequences are most stable in a right-handed double helix, which you'll be most familiar with. However, we know that DNA can form other structures. One such alternative structure is something called a quadruplex, which is formed when 1, 2, or 4 DNA strands combine together and create a quadruple-stranded structure. This quadruplex structure is formed by alternative arrangements of hydrogen bonds and is similarly stable and reversible, just like the double helix, although its construction tends to be more sequence specific. So the two most commonly known quadruplex structures are G quadruplexes, which are formed mostly out of guanines, and I motifs, which are formed mostly out of cytosines. So to summarize and give you grounding for what I'll be talking about later, DNA in its native form is typically present as 2 strands of bases arranged in an imposing reverse complementary fashion, but can sometimes form stable quadruplexes that are composed of 4 strands arranged in various ways. However, representing DNA as a chemical structure becomes very quickly limiting in terms of space and time, and confusing in terms of identifying the things that matter. One of the most common representations of DNA that geneticists use is a sequence of letters, A, C, G, and T, corresponding to those 4 basic building blocks. These letters are combined in a linear sequence that can be thousands to millions of bases long, a little bit like text in a book. We call the collection of DNA that resides on a single connected strand a chromosome, and a collection of chromosomes, which could only be one, related to a single organism, we call a genome. Representing DNA sequences in all their complexity can be something of a challenge. To be more explicit, visualizing that linear sequence of bases in a single succinct image is challenging. Now, I've toyed with the idea of converting these letters into colours, which gives me a little bit more room to describe long sequences. And also wrapping the coloured sequences into a spiral. But even a compressed spiral representation of a DNA sequence has its limits. For example, this works well for sequences that are 1,000 bases long, otherwise known as a kilobase. But I have trouble distinguishing individual bases when sequences get longer than about 30,000. So the figure on the left here, just to explain what you're looking at, is a spiral representation of a repetitive sequence that encodes for a protein called ubiquitin, which, as its name suggests, appears in a lot of the sequencing runs that I've done here. The figure on the right represents about 1% of the highly repetitive centromeric sequence of human chromosome 22. Even with this spiral representation, you need to squint to make out individual bases. And to drive the point home, the entire centromere of this chromosome is 100 times that size. And the entire chromosome, chromosome 22, is over 1,000 times this size. So if I want to show a representation of a single piece of DNA across its entire length, a spiral representation isn't going to cut it for the longest sequences. I need to find something better. So when thinking about how to visualize larger sequences, I started thinking about repeats in DNA. If I start with the assumption that DNA sequences are random, then I would expect each base to repeat on average about once every 4 bases in the sequence, or about 12 times in this 48 base sequence here. If I pair 2 bases together in a dimer, then I'd expect each dimer to appear on average about every 16 bases, or 3 times in a 48 base sequence. And so on, with the expected distance increasing by a factor of 4 with each individual base. As these fixed length subsequences get longer, the ability to distinguish the individual repeat sequences diminishes. But I realized that the location of the repeats is enough to identify what was repeated. In that case, I can use dots to represent the repetitive bits of a sequence. If my dots are too dense or too sparse, then I can just adjust the length of the sequences that I'm looking for to make images just the right amount of interesting. So I started thinking about how I would visualize that repetitive structure. But before I get into that representation, I want to talk briefly about what's come before and why I care about this. About 3 years ago, I managed to encourage Jodie Chandler, Amy Shepherd, and Tiffany Boucherie at our institute to extract some DNA from the rodent parasite Nippostrongylus brasiliensis. I've also tried to do this myself, but unfortunately haven't been quite as successful as they were. I ran the DNA that they extracted on our nanopore MinION sequencer, and at that time got only a sprinkling of sequences that were fairly high quality, but not nearly enough for what I wanted to do, which was assemble a genome. But I was curious about what I could find with what I had. So the Sanger Institute in Great Britain had previously produced a genome sequence assembly for Nippo using an Illumina sequencer, using DNA sequences that were about 100 bases in length. So I had a go at seeing how many of those Illumina sequences I could find in the nanopore reads that I had generated on our MinION sequencer, our much longer nanopore reads. This is a visual representation of a small fraction of that search using a free software program called Tablet. The mouse cursor here in this image is highlighting a single one of those Illumina reads as a red horizontal line, which represents a single sequence that came out of the sequencer. And these sequences, other sequences, are piled up on the top and beside all around there, so that it's possible to work out how many times an Illumina sequence matches the nanopore reference sequence that I'm interested in. The bases in the reference nanopore sequence appear in this plot as vertical lines, but you don't see all of the bases. It's difficult to see individual bases at this scale. What confused me the most when looking at this visualization is that I was expecting the Illumina sequences to hit my nanopore sequences pretty much randomly, which would produce a fairly uniform and flat profile. But what I got instead was a weird inverted mountainous profile, which you can see in blue at the top here. I actually spent and wasted a lot of time trying to write and use different programs to get rid of these things that I saw as visual eyesores. Now things got weirder for me when I tried matching the nanopore reads to themselves. This was something that I didn't initially expect to get any results from, because the number of reads that came off the sequencer was so few that the chance of seeing more than a few sequences that matched the same region in the genome was almost zero. I was surprised to see that I got a similar patchiness and coverage as the Illumina search, but it was even more pronounced and hard-edged in the nanopore to nanopore search. My discomfort was increased when I zoomed into the sequence, because in some places there were these chunks of different looking sequence that had different colouring, different base colouring, suggesting that there were a vast number of patchwork chimeric sequences that just happened to share bits with each other. I was really confused at what was happening here. And I realized that I should probably find some way to better determine what was going on, and how common this problem was. And in order to explain to others what I was seeing, I needed some way to visualize these things on a much larger scale. So in 2017, there was this Reddit user, Half-Eaten Scone, who had created and reported on a lyric visualization tool called SongSim. You put in the lyrics of the song, and it will visualize the repetitive elements in that song and the words of the song. This tool was actually based on existing plots that are frequently used for DNA alignment, which are called dot plots. So each dot or square in this matrix represents a comparison between two words in the lyrics of the song. In this case, Mary had a little lamb. Where there is a coloured dot, it means that the word is the same. In the example highlighted here by a lightning bolt, the fifth word is being compared to the 34th word, and that word lamb appears quite a lot in this song. There's a solid line down the diagonal because each word is being compared to itself and each word is the same as itself. Now this visualization was demonstrated about the time that I was looking for inspiration around how I could visualize repetitive sequences. So I was very interested in how it was done. What I noticed was that this approach concentrates on the words of a song rather than the letters. And I saw parallels between words and songs and fixed length subsequences of DNA, which we call kamers. Now this approach is very visually interesting and colourful. But I and others that I spoke to in my presentations, after my presentations, found this representation somewhat challenging to interpret. I wanted a visualization where the sequence would be represented as a single horizontal line across the screen with, or the image, with a dot showing up above it, indicating the position where a repeat of a chunk of that sequence could be found. So, based on that idea, I've created a program that I call REPAVER, which is an acronym for Repetitive Pattern Visualizer. I'm going to take you through the way that this program processes something called a tandem repeat, a situation where the same subsequence is repeated immediately again and again and again and again and again. In this instance, my repeat is 3 bases in length. A 3-base trimer with the sequence TAC, which for simplicity I have represented below as a magenta arrow. this sequence is present 5 times. So that first arrow is repeated 4 times, 3 bases away, then 6, then 9, then 12. But if I want to fully describe the repetitive nature of this sequence, then a one-dimensional display isn't good enough. The second copy is repeated 3 times, and the third copy twice, and the fourth copy once. And this is further complicated because the repeated fragment is 3 bases in length. So if I step along one base, then I get a sequence A, C, T, and that is also repeated. And then one base more, I get a sequence C, T, A, which is also repeated in a similar fashion. In addition, all of these repeats have what is essentially a mirror image. For example, the last sequence fragment has 4 copies that came before it as well. So I thought I needed another colour to represent these backwards repeats. So here's what that looks like when just the TAC portion of the sequence is represented in dots. This visualisation here is a REPAVER representation of a short tandem repeat. But there's more. When we look at a bunch of letters on a computer screen, we tend to imagine it on its own, unmoving and alone. But you may recall me talking previously about double-stranded helixes and quadruplexes. So I want to add to that by explaining what it means for a sequence to be repeated in DNA. Now, I've told you that DNA is almost always double-stranded, paired up with the reverse complement sequence on the other side. If there's another reverse complement sequence floating around somewhere nearby, and the only thing holding one sequence to the other is hydrogen bonds, then I think that those bonds could probably fairly easily shift over to the other sequence. My current working hypothesis is that a quadruplex-like structure would be formed from 2 double-strand subsequences that is stabilized by oscillating hydrogen bonds. I am not completely sure how I would test this at the moment, but please offer me your suggestions if you have any ideas. But if you consider that DNA is a physical object in 3D space, then it might be easy to see that a sequence could be physically reversed without breaking anything in the sequence. As I said before, this would probably cause strain on the structure. But I think it's useful to have an idea of this to know if it's happening at all. So the combination of complementation and reversal gives us 4 different ways of seeing whether a short sequence could potentially be paired up with another sequence that's floating around. When I'm looking for repeats in DNA, I think it's a good idea to look for all 4 of those possible configurations rather than the basic Ford repeat. So, going back to that same sequence, here's a quick demonstration of the full 3-base trimer repeats diversity from the sequence that I showed you previously, where you can see that, that TAC repeat structure in the middle. But there's also a whole bunch of other patterns in there which are not necessarily obvious from looking at the sequence alone. As I mentioned before, we call the collection of DNA that resides on a single connected strand a chromosome. Now, chromosomes have an impressive range of lengths in the natural world, varying in size from a few thousand bases to a few billion bases in plants. Representing this massive dynamic range is challenging. I've come up with my own workaround, which was actually based on me looking at the reflections of streetlights on water and thinking, I want something that looks like that. The top image here is the COVID-19 viral genome, which is a little bit under 30 kilobases in length. And from my perspective, it looks pretty random. All of the 4 different repeat types that I was interested in appear in roughly equal proportions. The bottom image is the longest chromosome found in people, chromosome 1, and is a little bit under 250 million bases. It's very much not random. What I've noticed is that as I look at longer and longer stretches of DNA, beyond a few 100,000 bases, I start seeing common repetitive patterns. In the range of a few 100,000 to a few million bases, the start and end of each non-repetitive chunk will commonly have complementary sequences. So you could say that the start of a region is like a reflection of the end of a region. Stretching out a bit further to longer sequences, I tend to hundreds of megabases, I start seeing an abundance of very long tandem repeats, shown here in magenta. And these repetitive chunks seem to act as maybe sturdy supports to a collection of non-repetitive regions, grouped together based on some loose connections that are not always obvious to casual observers of the genome. So the collection of chromosomes that make up the genetic code of an organism is called a genome. A DNA sequencer can, at best, produce a fragmented version of that genome from many different cells. And it's a technically difficult task to assemble those pieces back together into separate chromosomes. I find this REPAVER representation to be very useful in identifying regions that could give the assembly programs grief. For those who aren't familiar with genome assembly, you typically first identify ideal fixed-length subsequences or seeds for matching bits of different pieces of sequenced DNA. The ideal seeds are ones that have high complexity and are easy to identify in other sequences. There's a bit of an art to getting a good assembly. You need a good eye for the repetitive structures that can cause problems, including the things that look locally unique, but are duplicated in completely different regions of the genome. If you're lucky and have control of the sample preparation, long fragments can be sequenced to make sure that none sit entirely within repetitive regions. So there's always a unique bit in each sequence that can be matched to neighbouring sequences. Now, people have frequently compared genome assembly to jigsaw puzzles, and I see photo stitching as basically a jigsaw puzzle where you get to decide in advance what the pieces look like. Even though it's one-dimensional rather than two-dimensional, genome assembly still has the usual issues. Sometimes it's hard to know which way round a sequence goes, like with ropes and poles in jigsaw puzzles. Sometimes you get bits which are complex, but highly repetitive, like windows and bricks. And sometimes there are low complexity regions where the same thing is repeated in close proximity with itself, just like the sky and the grass. Over the past few years, I've been running my REPAVER visualization program on the longest assembled genomes that I can get my hands on. It's not uncommon for me to look at something that the authors claim to be a multi-megabase chromosome scale sequence, but it has an almost complete absence of tandem repeat regions. That seems to be a very common feature of most chromosomes. But we're getting there, and quite rapidly. You may not be aware that the human genome has never been fully, completely assembled. However, there's a group working on that. The Telomere to Telomere Consortium, led by Karen Meager and Adam Philippi, has produced a new draft version of the genome from a human hydatidiform mole. And pretty much the only missing bit of that assembled genome is tandemly repeated ribosomal DNA. They know exactly what's missing. They're just not quite sure how many copies there are. So I'm eagerly looking forward to what the next few years of genome assembly are going to bring us. So I've been doing this work, as I said, as a side project, but I want to specifically thank Jonathan Eubank, Graham LeGros, and Tiffany Boucherie for starting me off down the rabbit hole of repetitive DNA in the rodent parasite Nippostrongylus brasiliensis. I also want to specifically thank Dr. Anne McCartney, who on multiple occasions has successfully run my visualization programs and given me ideas and suggestions for improvement. If you want me to have a look at your pet DNA sequences, feel free to contact me, bioinformatics@grindgene.org or grindgenebio on Twitter. Thanks for listening. Ngā mihi nui, kia koutou, kia ora.
Yeah, I have some one comment. So you are would like to look at quadruplex of DNA, but in the nucleus, DNA are never naked and are always, at least, interacting with protein and at least histone and other proteins. So I think it's in vitro, it has been postulated that some factor like HMG, high mobility group box transcription factor can bind quadruplex of DNA. But yeah, in nature, it's quite difficult to find those. So the only way that maybe you could look at is all those people looking at the genome structure and at the 2D that you are looking, but not the 3D structure of the genome. Did you think about applying your visualization to the 3D structure of the genome?
I have tried to think about that. One of the things that my algorithm can pull out is an indication of the sequences that are present here that are the most repetitive. So I can look at this and see what's the most repetitive reverse complement structure. And if you're looking at visualization, you might think about creating a probe for that sequence that fluoresces when it binds to that sequence. And so you might put that in a cell and have a look at the 3D structure of the genome based just on that sequence. Currently, people use a process called Hi-C, where they, I think they break up the chromatin and use that to attach distant pieces in the genome together to find out DNA sequences that are close in three-dimensional locality, but far away in terms of the genome sequence. One of the issues with high C that's most commonly used is that it uses Illumina sequencing for identification. And if you see this assembly here, I'm looking at subsequences of length 100 bases, and that's similar to the length that comes off an Illumina sequencer. And these are things that are repeated all over the place. And so I don't think there's any way for high C to be able to detect when this is happening. So it's going to be quite challenging for me to be able to link up the observations that I'm seeing with the high C that's traditionally done. There is a technique that uses nanopore sequencing, which is a similar process, and they call it pore C, and that allows you to combine adjacent three-dimensionally adjacent structures into nanopore reads, and so that might be a better thing for me to use to see if these repeats that I'm seeing actually represent close proximity in 3D space, but I haven't got around to looking at any of the pore C data that's lying around to see if there are any similarities with what I'm seeing here.
Yeah, I was wondering that you don't see any tandem repeat arrays, which I'm a bit surprised because Genomes can find a lot of like STR markers in the chromosomes, so why isn't there any?
Yeah, so there are actually some STR repeats in here. So that's these green blocks at the bottom. But yes, the lack of long tandem repeats, which we see quite obviously in the telomere to telomere consortium chromosome assembly, I think is a side effect of the way that this genome has been assembled. This was assembled using Pacific Biosciences SQL sequencer and Illumina reads. And those reads only go up to about 10 to 20 kilobases. And these long tandem repeat arrays that I see, they span regions of the orders of hundreds of kilobases to millions of hundreds of thousands of bases to millions of bases. And so most of those Pacific Biosciences SQL reads are going to sit within those tandem arrays, and because there's nothing non-repetitive around them, you can't place them anywhere in the genome. So all of those repeat regions get collapsed, and I think that's what's going on here. But it's been quite a struggle trying to convince people that there might be something missing in these PacBio assemblies.
Have you looked at this in the context of disease, of things like Huntington's, where you do have repeats? Is there any kind of correlations with any diseases? Have you looked at that?
Short answer is no. Mostly I've been working on visualizing these things to see that they're happening, and yeah, it's another step down the road trying to understand why they're happening and whether or not there's a relationship between disease and the extension of repeated structures. I mean, yes, we know that there's a lot of repetitive structures and structural variation, which is correlated with disease. And I think and hope that this method could be used to better work out when that's happening. But I'm still at the stage where I'm just trying to get out this visualization and show it to other people. Yeah. Okay.
Thank you.
Thank you.